Here is the second video, the last video for Chapter 17. We're going to go through some examples of serological tests that are used in clinical diagnoses. First, though, let's talk about monoclonal antibodies. First, let me tell you what they are. And let me set the stage. Normally, a B cell, a particular B cell, B lymphocyte, synthesizes one type of antibody, an antibody to an E. coli, like an IgM, or an IgG to some other kind of organism. There is a huge population of different kinds of B cells in an organism, and lots of different kinds of antibodies that are produced in response to the various antigens that the organism has been exposed to. But to be useful as a tool, biologists need large amounts of a single antibody, and that antibody alone. Therefore, we need a method to culture a population of B cells derived from a single kind of B cell, so that this population of B cells would allow us to harvest a single kind of antibody, very specific, pure antibody against one particular epitope of an antigen. This population of cells would be correctly described as monoclonal, and the antibodies made by this population of B cells and their plasma cells are called monoclonal antibodies, or sometimes you just see them written out as MABS, monoclonal antibodies. In contrast, excuse me, antibodies obtained from the blood of an immunized animal are called polyclonal antibodies because of the variety of antibodies made. They have a variety of academic, medical, and commercial uses. Some examples. Antibodies are used in several diagnostic tests to detect small amounts of drugs, toxins, or hormones. For example, in the pregnancy test kit, you look for HCG, or human chorionic gonadotropin, in the urine of a pregnant woman. Another diagnostic test use for these antibodies is diagnosis of AIDS in the ELISA test. So, test kits, things like that. These monoclonal antibodies may be used in the detection of and therapy of cancer. So you can make very specific antibodies against cancer cells for their destruction. Monoclonal antibodies have also been used to treat viral diseases, particularly ones that are very tough to treat. Monoclonal antibodies can be used to classify and identify strains of a particular pathogen. Also been used to reduce the problem of organ rejection in patients who've had organ transplants. Okay, so how do you make them? Let's say you want to make a monoclonal antibody against hepatitis B protein in the capsid structure of the virus. You take that antigen, you inject it into a mouse. You see that on the picture here. Lots of the B cells of the mouse will be activated, producing a variety of antibodies, polyclonal antibodies. You kill the mouse, take its spleen, separate out the different B cells. You add those B cells to cancer cells, not cancer cells of humans, not anything to do with that, but cancer cells that can be grown in culture called myeloma cells. And the cancer cells and these turned on activated B cells will fuse together, forming cancer cells that now will be making pure antibody against one particular antigen. That's called a hybridoma. You separate out these hybridoma cells. You find the particular B cell cancer cells that are making the antibody against the antigen you're interested in. You purify them, you grow them, and you now have large amounts of pure antibody that you're interested in using. I've got two examples of test kits here. These are both for pregnancy. Here's an example of one of the enzyme immunoassay tests, comparable to what you find in a pregnancy test kit. And this has got monoclonal antibodies in it that will react with antigen in the patient's urine. Okay, purposes of immune testing. Is it to detect antigens of a particular infectious agent using known antibodies in your test kit? Or is it having a test kit that's got the antigen, a known antigen, and you're looking for the possibility of antibodies in the patient's blood to a particular pathogen or antigen? Well, you can do it both ways, both the purposes. You can use this type of testing to detect antigen, or you can use this type of testing to detect antibody in the patient. So I'm going to take you through some of the tests. Some are more commonly used these days than others, and you're going to be probably familiar with some of them. Okay, in an agglutination test, you've got a large antigen. Antigen is Ag. You add an antibody. Again, you could be looking for an antigen in the patient using a known antibody, that you could buy, not at the grocery store, but from a supply house of antibody. Or you could be looking for antibody in the patient, and you're using a bought antigen. Either way, doesn't matter. These antigen-antibody complexes are large, and when they occur, you can see them visibly, for example, on a slide. You've probably seen this before if you've had A&P and you've done blood typing, because that's an agglutination test. Remember, look at the card at the bottom left. You see a blood typing card. You add the blood of the patient that you're interested in typing, and you have antibody that is added, like anti-A, anti-B, anti-RH, and you mix them together on the slide, and you look for clumping of the red blood cells. And you can see it in the first well where anti-A has been added, meaning that the person's blood has A markers, agglutinogens on them, or antigens. And you can see it in the third well where RH has been added. That's the anti-D antibody. That means that the D antigen, what's called the RH antigen or agglutinogen, is on the patient's blood. So this is A positive blood. You're using a known antibody to find particular antigens. The test on the right is a monospot test that we're going to be doing in lab also. On the left, you see agglutination. Looks like little fine particulate matter kind of settling out. Again, in this case, you're looking for antibody in the patient, and we use a known antigen of the Epstein-Barr virus that causes mononucleosis. It's called the monospot test. On the right is just a kind of a mixed patient's serum without antibody added to the antigen, and you get just kind of like a milky, homogeneous solution, but no agglutination. We're also going to be doing a latex bead agglutination for staph identification. That's another agglutination test. So they're used a lot. Okay. Here's another example of agglutination. This is called a hemagglutination test. And in this situation, again, it's you've got an antigen and an antibody that react together, but thrown into the mix are red blood cells, which give you a much more apparent reaction. You get a binding between the cells and the antibody-antigen reaction, and because of the red color of the cells, you get a much more apparent type of obvious reaction that's much more visible. And in addition to the hemagglutination reaction, you can do a hemagglutination inhibition reaction. This has been set up in a microtiter plate showing this is a test for indication of flu, a person having flu. Okay, here's a second test called a precipitation test. It's kind of similar to the agglutination test. Difference is that your antigen is very tiny and it's soluble. There are different examples of how this is used. You still get a kind of a visible line where the antigen and the antibody come together. It's not used all that commonly much anymore because now there are more sophisticated tests, but they've used this a lot in identifying hormones and certain types of drugs that are in the patient's serum. It's kind of like a variation of a agglutination test, but not as obvious. Okay, here's a neutralization test. Let me give you the example of a botulinum toxin test. Let's say you have a can of food, and the can is swollen, and of course everybody would, I would hope, think of botulism, because the botulinum bacteria, Clostridium botulinum, makes a lot of gases, and it often causes the can to swell. Now, one of the ways they verify the toxin is by injecting it into mice. Now, mice, when they come in contact with the toxin, would die. But of course, the mouse could die of other things in the food. Doesn't necessarily have to be the toxin, or maybe you break a blood vessel when you inject the mouse with a syringe and it dies from blood loss. It doesn't have to be the toxin. So what you do is you inject the mouse with the toxin, and then you give a very specific antibody to that toxin, and if the mouse does not get ill, then it shows that the antibody has neutralized the toxin. That's why this is called a neutralization test. It's also used on viruses in addition. And that's what you see on the picture, is the antibody neutralizing the viruses. Okay, here's a fluorescent antibody test. This is used very, very commonly. You can do it a couple of different ways. In either way, you're using a fluorescent dye. And that fluorescent dye really lights up in a microscope, and you can see it very easily. So these dyes are linked to an antibody, and so if you look at the direct picture on the left, where it says identify antigen, right here, you see that the fluorescein dye has been labeled onto an antibody. You add it on a slide where the antigen has already been added. Again, you don't know if this is the particular antigen to this antibody. Again, you're trying to find out if the antigen is present. Let me give an example. Let's say you're looking for rabies antigen in the brain of a rabid, or what you think may be a rabid dog. So you've put the brain cells of the dog onto a slide, that's your antigen, with potentially rabies virus. You add a special antibody made to the rabies virus that has a fluorescein dye attached to it. When the antibody attaches to the antigen, if that rabies antigen is there, the fluorescein dye lights up in a microscope, would be a fluorescent microscope using UV light, and it's very easily seen. And the picture you're looking right below at here, this is what it would look like, although this is not against rabies, it's against Legionella. And they make fluorescein dyes in different colors. You can also use this type of test to identify the patient's antibody. So in this case, you add a known antigen. This time you're not looking for the antigen. You've got the antigen in your hand. You add it to the slide. You then add the patient's serum, which may or may not have the antibody. There's the antibody, potentially, in the patient's serum. Then you add another antibody against that antibody, and it's got that fluorescein dye on it right there. If the patient has the antibody to the antigen you're looking for, you'll get a light up of this fluorescein dye. So again, two ways to do it, direct and indirect. So that's the fluorescent antibody test. I mentioned rabies. They also use it a lot in herpes. Okay, here's probably the fastest growing group of tests right here. The enzyme immunoassay, or EIA. In this case, again, you can use it to both identify the antigen or to identify the antibody in the patient's serum. Let's say that you are looking for HCG antigen. It's a hormone in pregnant women's blood and in their urine. If you're pregnant, you got it in your urine. So this is a pregnancy test kit. You would have known specific antibody to the HCG that is placed onto paper. I've got on here that it's put onto the surface of something. It could be added to like a well, like a plastic well like this. This thing is a little plastic well right there. Or you could put it on the paper, like inside of these little test kits right there. And this antibody is attached to an enzyme. When you add the person's urine, if they have HCG, there is a reaction between the antigen and the antibody, and this causes the enzyme to facilitate a reaction. I should mention that there's a substrate thrown in, already in there on the paper, for the enzyme. And when the antigen and antibody comes together, the enzyme then gets activated. It acts on the substrate. Substrate changes colors. So, of course, in a pregnancy test kit, you get like a little pink line, little blue line. That's what's going on. The enzyme is activated. It acts on the substrate. Substrate changes colors. None of this happens if there's not HCG in the person's urine. Again, the antibody's already on the paper. Now, the pictures you see here, examples of, this is actually very, again, I could have put a pregnancy test kit here. It's the same kind of thing. But instead, I've got an example of the OroQuick, which is for HIV diagnosis, that you can actually buy over the counter now or online. And on the left is a similar type of EIA test for malaria. In the case of HIV, another way to do it is with these little plastic wells. So, for example, here is an HIV antigen, the blue thing. It's coated onto the sides of a little plastic well, like that. You add the patient's serum. The patient's serum is going to be this right here. Now again, we don't know if the patient has anti-HIV or not. Let's say they do. That antibody then attaches to the HIV antigen right there. Then you add another antibody. This is an antibody made against HIV antibody. It's antibody to antibody. It's got again, an enzyme and a substrate right there that have been added to the well. When that antibody reacts to the HIV antibody, you get a color change from colorless to colored. Here's another example of what would be done for HIV. Again, these are little plastic wells, and if you look at this apparatus that she's got here, she's doing, this woman is doing multiple inoculations, and this little thing she's got right there is one of those little, it's about a 4x6 tray, plastic tray. It's got about 96 wells in it, and so you can do 96 reactions in the well. This particular test is called the ELISA. It's a type of enzyme immunoassay. And ELISA stands for Enzyme-Linked Immunosorbent Assay. Again, it's a type of EIA. I think one of them got skipped. Looking for the complement fixation test. Oh well, I'll talk a little bit about that in class. Not used much anymore anyway. Here's the Western blot test. This is another test, most often probably used for HIV, but it can be used for other things. That's what it's famous for, though. The ELISA test for HIV antibody is the standard test done anytime you go in for HIV testing. And anybody can do that test. Well, I mean, you have to be trained, but you can train anybody to do it. And it's a pretty fast test. But there's some false negatives and some false positives. The Western blot is the gold standard. So if somebody comes out positive on the ELISA test, then you do the Western blot. And if it comes out positive, it's real. So it uses some techniques we've talked about before, like electrophoresis. Again, you are using patient's serum to see if they have antibody. You're using known HIV proteins, antigens. You separate them out on the in the gel. You do a blot, which transfers the fragments of the protein from a gel to a piece of paper. When I mean paper, I mean nitrocellulose or some other paper, not just regular old writing paper, for stabilization. And then you add patient's serum. And if there is specific antibody to these HIV proteins, you get fragments, which you can see right here. Okay? These are the antigen-antibody complexes, and you can visualize them. You can see another example over there. Again, much greater specificity, but takes longer, much more expensive. You have to be trained to do this. It's kind of a complicated process. And again, another look at a blot. So those are the key tests, and I've covered most of those, and that's all to that.